Here I'm going to share with you how you can use the projection tool on gEAR to identify cell types in your personal single cell or single nuclei datasets. First, you need to identify a list of marker genes in a specific cluster that's unknown in your dataset. Once you've identified these markers, you'll then need to create a gene list by clicking on the Gene Lists button to the left on the main page of gEAR. Once we're in the Gene List Manager, you then click Create New Gene List. You can upload either unweighted gene lists, which do not have quantitative data assigned to each gene, or you can upload weighted gene lists that do. Once you've uploaded your gene list and given it a name, you then go to the projection tool to the left. Next, you can select your gene list at the top left, as well as the data set that you want to use for analysis. Here I'm using a set of genes that are specifically expressed in outer hair cells. However, if you want to identify an unknown cluster in your single-cell RNA-seq data set, you're going to want to use a list of marker genes that are specific to that cluster. You can also select the algorithm at which you want to do this analysis. And here we're going to use the values from principal component analysis. So what happens next is gEAR will use transfer learning to identify the cells whose transcriptional profile matches your gene list the best. And so as I mentioned, this gene list here is specific for outer hair cells. And so what we would expect to see is high expression in the outer hair cells. Additionally, if you scroll down, you'll see this expression mirrored in the UMAP projection. And lastly, you would see it again, except this time in the form of a violin plot. Welcome! The gEAR team is happy to announce our new support for spatial transcriptomics datasets. Our team has generated a spatial dataset by applying the Visium HD platform to adult mouse cochlear sections. So let's jump right in. At the top left search bar, we can enter in our favorite gene. Mine's PAL4F3, a known pioneer factor expressed in both inner and outer hair cells. Once you click on the search icon to the right, we initially see a panel below that shows 3 images. The first is image data acquired from a microscope. In this experiment, we used a bright field microscope to capture H&E stained tissue. In the next image, we see areas in which PAL4F3 is expressed, with the bins that have highest expression in dark green. The last image shows cell type annotation that's been uploaded by the user. If no annotation is provided, then unbiased clustering will be included. Within this panel, you can also filter the binned regions shown based on the total number of genes detected in a given area. For this analysis, we are using a cutoff of 200 genes. And so as I mentioned before, PAL4F3 is expressed specifically in hair cells, and at this magnification it's very difficult to identify the regions in which PAL4F3 is expressed. So what we can actually do is zoom in by highlighting our region of interest, and in the panel below we then see that highlighted region zoomed in. We can also further filter the cells at which we're interested in by clicking on them at the right. And so here I've removed interdental cells, mesenchymal cells, and this group that we've called other cells. The next panel shows a UMAP projection that correlates with the spatial data above. Here you can highlight those cells that you're interested, just as before. However, when you click, it focuses only on the cell type that you're interested in. Our last panel demonstrates cell type specific expression of your gene of interest, and it's shown through a violin plot. And as you can see here, expression of PAL4F3 is specifically in inner and outer hair cells. Moving forward, our team is committed to generating reference datasets using spatial platforms spanning from embryonic to adult ages of mice. It's our hopes that the auditory field can use these datasets to bolster their transcriptional studies by comparing cell annotation with their own personal datasets. Perhaps they can describe regional changes in expression not previously seen, and identify novel cell-to-cell interactions within the cochlea. We're so excited to see the unique and new ways that gEAR can be used to help you with your studies. Next, I'm going to demonstrate how the transcriptional information from spatial datasets can be extracted and analyzed using various bioinformatic packages. But first, you'll need to download your spatial dataset of interest as an H5AD file by clicking on the three dots at the top right of the panel. You can then scroll down the drop-down menu and click Download H5AD. Once downloaded, you can extract the data using a number of computational environments. In my case, I used RStudio and the Seurat package to integrate my single nuclei dataset with gEAR's adult spatial dataset. This resulted in the accurate clustering of many cochlear subtypes between the single nuclei and spatial dataset. It's our hope that as we generate more spatial datasets, these can be used not only for accurate cell annotation, but also for identifying where cells in a single cell dataset are located in the cochlea.